My name is Scarlett Abou. I am working in the Institute of Human Genetics in Montpellier. And today I will talk to you about the changes in my life when I start to use Voyager pipettes. Because actually I'm working with patient samples and we are trying to manage a lot of samples, and it was time-consuming to use only manual single-channel pipettes. And when the Voyager appears, I tried first and I was convinced about the use, especially because it makes my experiment faster and less harmful. For the loading, it was really loading in the gel, into the gel, it was really huge because as I told you, 40 minutes against 5 minutes. It's unbeatable, we can say that. My colleagues that were around me were curious and they wanted how maybe I can use them also for loading qPCR samples in 384 plates. And sometimes we were like fighting, okay, do you need this pipette?
I am Gaël Petitjean. I am a research associate in the team and I work on HIV-1 infection for almost a decade now. I work on different aspects of HIV-1 infection and all the interaction between HIV and the cell host. For our purpose study, we have a lot of applications that come from qPCR to the amplification of the HIV-1 genome at the single molecule level or in single cell level, and that's why we have a lot of applications that need high throughput. Before trying the Voyager, we worked with monochannel pipettes and it was quite difficult. We spent a lot of time doing the experiments, so that it was a critical point for us to be able to have a high throughput, and that's why we choose to work with Voyager and INTEGRA, and it was a real gain for us. With a single channel pipette, we spend a lot of time by loading plates or gels, mostly 1 or 2 hours, and now with the Voyager, we save a lot of time. We only need a few minutes, maybe 10, to load a plate or gels. It's quite faster.
So my name is Suzy Tenaucier and I'm a research assistant in the molecular virology lab. So I'm using the Voyager pipette to do qPCR analysis. So before I was loading a 384 well plate with 1 tips per well, and now with a Voyager I can do 8 well in one time. So I changed for time saving because I'm loading a plate in 15 minutes compared to 1 hour with a monochannel pipette, and also for reproducibility in the results.